introduction. Thank you for the visitors for bringing the rain. It's nice to see that again. We haven't seen that in a long time. On to more relevant things. I'm going to tell you about Vista Therapeutics. We are integrating nanotechnology and biotechnology and engineering to help improve diagnoses and treatment of humans as well as animals for almost any kind of health condition. Today I want to emphasize that we are about to launch our first platform, which will include both disposables and a sensor that will allow for continuous biomarker measurement, as well as rapid, frequent measurement. What are the unmet needs and problems that Vista's technology can address? Well, let's first make a few descriptions of what we're talking about. We're talking about biomarkers. Biomarkers are molecules that are made by the body in response to stress to try and either repair the damage, to prevent further damage, and in the case of an infection, to get rid of whatever the infectious agent is. There are lots of different kinds of biomarkers. We're going to focus on protein biomarkers. In the case of injury or rapid onset disease, there are several biomarkers that are rapidly induced or repressed almost instantly. The various squiggly lines represent the speed at which these change. They begin to change literally within minutes of the inducing event, not hours. And they present a wealth of information, but the way they're measured today, we can't take advantage of that information. For one thing, the sample size is way too long. It was way too large. 15 mLs of blood makes it unrealistic to continuously measure anything. Secondly, it takes a long time to get the results back. 4 hours is rapid. And if we're talking about something like acute myocardial infarction, 4 hours is irrelevant ancient history. And that's when you get the information. If you're lucky, is 4 hours later. But sometimes it can be a couple of days, as you know. So in fact, the way that doctors measure biomarkers today, we miss 99% of the relevant information. It's sitting there, it's available, but it's not utilized. They can't utilize any of the trends to identify if you're getting better or getting worse. You can't identify peaks to determine if you're at where you are in the progression of the disease. So the obvious solution to this is to be able to continuously, or at least very frequently, monitor changes in biomarkers as they are occurring, not present some historical tone, pun intended, as is done now. Vista's technology will allow that kind of continuous and very rapid measurement. Let me give you a couple of examples. One for continuous measurement, one for snapshots. This is just one of many examples. Blood trauma happens in car accidents. If you're visiting certain foreign nations, when explosions occur, lots of different causes for blood trauma. Right now, you're admitted, if it's severe enough, into the ICU. The doctors begin to monitor you. They look at traditional measurements like creatinine clearance, urine output, heartbeat, temperature, etc. And it takes, frankly, up to 20 hours before you even begin to physically manifest the worst possible outcome of blood trauma, which is multiple organ failure. By the time you've identified that this patient has multiple organ failure, it's generally too late and the treatment options are very limited and the outcomes are frequently quite unsatisfactory. Vista's technology would help solve this problem in a very specific way. Namely, we would connect that patient. There would be our system at the bedside of every single ICU and ER patient. We would connect that patient to our biomonitor and continuously measure whatever the set of biomarkers is that's relevant for that particular disease or injury. These biomarkers, as I indicated, begin to change along with the event. And they're not just correlated to the event, they're causative to the event. So if we can interfere with increased production, say, of pro-inflammatory cytokine, we can reduce that inflammation. This provides doctors with a great deal more in the way of treatment options, and the outcomes are generally much more positive. Another very powerful application is very rapid snapshots. So any time where there are a large number of people that need to be screened under potential epidemic situations where the vaccine is limited, you want to give the vaccine to the correct individual. Well, our technology can be applied to determine if you carry the host vector, what your response is going to be, and from start to finish, one can have the answer in 5 minutes. So both continuous measurement of biomarkers and very rapid measurement. So how do we get away with such sensitivity? And I want to emphasize the sensitivity. If you stick a pencil into a glass of water, the drop that will fall off is somewhere around 100 microliters. We can measure 100 molecules in that drop, which represents about 10 to the 18th molecules. That's 6 to 8 orders of magnitude more sensitive than the most sensitive testing system on the planet today. So, biomarkers go up. The way we measure them is we've created a nanoscale field effect transistor. All that means is when the charge near the surface changes, the current running through the nanowire changes accordingly. We stick antibodies onto the surface. Proteins in the blood all carry a charge, otherwise they would not stay soluble. When they bind to the antibodies, they're always binding and falling off and binding and falling off. When they bind, the current running through the circuit increases. So it's an electrical detection system. How do we get away with using such tiny samples? Well, the nanowire, this is a scanning electron micrograph. You can barely see the nanowire down at the bottom. And there's a cartoon of a couple of antibodies. That is on the order of 100 atoms across. We can put many of those circuits onto a chip. The working part of that chip has 180 circuits, so we can measure 180 different target molecules in an area the size of a grain of rice, and we need a vast flow rate of 3 microliters a minute. If we started today pulling that out, it would take about 4 months before you bled to death at that rate. So, just in summary, this technology will help usher us into the new world of personalized medicine because the doctor can actually watch the results of his treatment while he's making that treatment. So where is Vista in our commercialization process? We have kind of a microbrewery, handcrafted pathway for manufacturing our products, which include disposables, sample preparation kits, as well as the nanocart themselves. We'll end up selling initially tens and then hundreds and eventually thousands of nanocards per instrument. All of these are now being manufactured. You can come and see them working at our booth. We need to put them under one roof and put all of the people who do this work in the same location versus being spread out across the country as we are now. Our initial markets will be regulatory free, and they'll be focused in the pharmaceutical sector. They have great needs for doing all of this kind of testing, this rapid kinetic testing on new drugs. They can use it for clinical research, developing drugs for any kind of highly dynamic disease, again, like a heart attack, and clinical trials. It's a significant opportunity, about $6 billion globally, but the holy grail is when we achieve FDA or foreign country FDA analog approval and can stick our machine on the bedside of every single ICU patient. That's a $20 billion global opportunity. What is our near-term strategy? Well, of course, we have to raise some money to start the fabrication and to sell the product. We need to build our core fabrication center and hire the key sales and fabrication people. We'll start by selling a service that's a bit more forgiving of errors. Then we'll sell on a strategic basis to pharmaceutical companies who are working on acute disorders. So we'll initiate strategic deals in about 4 months, and we'll start looking for a seasoned and experienced partner to help us get through the FDA process, somebody like Medtronics. Our financial projections, there's an error, there should be an extra 0 on the x-axis numbers. This is very conservative, about $30 million in gross revenues 3 years out, with more rapidly rising net revenues simply as an economy of scale for making semiconductor products. We expect to start selling our products 4 months from financing, close of financing. We'll start seeking approval from the FDA in about 10 months. We hope to become cash flow positive within the first 18 months. And we will just simply, I'll say here, we'll be very opportunistic about looking for a new home. Analogous companies have just sold to Medtronics for $800 million. Finally, who are we? I have started 3 companies, took 1 public, it's still in business. Second company we just sold to Genzyme. A 3rd company, I think I saw Michael Schaefer here someplace, got off to a roaring start, kind of fizzled at the end, but it was a first of its kind, a very powerful company. Charles Lieber, besides being nominated for the Nobel Prize the last 3 years in a row for this technology, is also the co-founder of Nanosys, the pioneer in nanotechnology. And our 2 senior manufacturing people and engineers, Noah Clay and Ron Selesky, are ready to join us if we can afford to have them here. Ron is here today. The take-home lessons, and then I'll shut up. Basically, we are going to, he said modestly, rule biomarker measurement. There's a large demand for our products right now. Biomarker sales are already at $7 billion annually. We have a recurring revenue stream model. Enormous markets, $26 billion. Our current focus is not R&D, it's to start selling products with your help. Start selling products. We have an experienced team. We've made most of the obvious errors. There's always room for new errors, but we won't make the simple stupid ones again. And a very strong IP position. We've got a great booth here, and you can come and see all of this stuff at work. We also have labs in Santa Fe, so while you're in town, you are welcome to come up and visit our labs in Santa Fe, and I'm going to stop here before the book comes out. Thank you for your time.